Thinkerflix.
DNA, genetics, epigenetics, and the theory of evolution. As citizen of the modern world, you probably know that deoxyribonucleic acid, more commonly known by the acronym DNA, is the molecular substance of genes, that the sequence of nucleotides in DNA determines the genetic information carried by a gene, and that the theory of evolution in its current form started with the now famous British naturalist Charles Darwin. During his voyage on the HMS Beagle from 1831 to 1836, Darwin observed species that seemed to be adapted to their environments, particularly in the Galapagos Islands, and began to question the prevailing belief that species were immutable. After gathering evidence for 2 decades, he published On the Origin of Species in 1859, detailing his theory of evolution by natural selection. Alfred Russel Wallace, a fellow British naturalist and explorer, whose extensive fieldwork in the Amazon basin and the Malay archipelago provided him with insights into species distribution and variation, wrote an essay in 1858 outlining his evolutionary theory and sent it to Darwin for review. This prompted the joint presentation of both men's findings to the Linnean Society of London in 1858. Today, evolution is considered a comprehensive framework explaining the diversity of life on Earth. It posits that all species share a common ancestry and have diverged over time. Terms such as natural selection, genetic drift, mutations, and gene flow are often used to describe the processes at play. This modern synthesis integrates Charles Darwin's 19th century concepts with Mendelian and contemporary genetic knowledge. To illustrate how science works by thoroughly examining empirical evidence for proposed theories, let's take a step back to a time before Darwin's theory and briefly examine an earlier theory of evolution that was discredited in its original form, but of which some parallels have resurfaced in the context of the more modern field of epigenetics, which we will examine later. This pre-Darwinian theory, Lamarckism, named after its originator, French naturalist Jean-Baptiste Lamarck, is an early evolutionary theory proposing that organisms can pass on traits acquired during their lifetime to their offspring. Lamarck suggested 2 main mechanisms. The first of these, use and disuse, proposed that organs or structures become enhanced or diminished based on their usage. For example, he theorized that giraffes' necks became longer because they stretched to reach higher foliage. The second mechanism, inheritance of acquired characteristics, proposed that traits so modified or developed during an organism's life, such as the longer neck acquired by stretching, could be inherited by its progeny. Lamarckism was eventually discredited for several reasons. There was a lack of credible empirical evidence, because experiments failed to demonstrate that acquired traits could be inherited. For instance, August Weismann's experiments, where he cut off the tails of mice for multiple generations, showed that tail length in offspring remained unaffected and indicated that acquired characteristics were not inherited. As we'll see later, emergence of Mendelian genetics clarified that inheritance is based on genetic information encoded in DNA, which remains unchanged by an individual's experiences or behaviors during their lifetime. Modern biology's understanding of DNA and chromosomes has established that genetic information is passed through DNA sequences in chromosomes, and these sequences are not altered by an organism's activities or environmental interactions during its life. And finally, Charles Darwin's theory of natural selection offered a more robust explanation for evolution. It proposed that random genetic variations that confer a survival or reproductive advantage become more common in a population over time, and is supported by the transmission of identified genetic material, notably DNA, from generation to generation, without the need for the inheritance of acquired traits. In summary, while Lamarck's ideas were pioneering in suggesting that species could change over time, the mechanisms he proposed for such changes were not supported by scientific evidence and were superseded by the principles of genetic inheritance and natural selection. The modern theory of evolution, as understood today, is not merely a body of knowledge or set of facts. It also defines a mechanisms and processes for how evolution occurs. Evolution cannot be understood without properly understanding these processes, which operate at the gene, individual organism, and population level. Let's start with a basic understanding of DNA, or deoxyribonucleic acid. As far back as 1869, Swiss chemist Friedrich Miescher first identified a substance he termed nuclein within the nuclei of human white blood cells. This material, now known as DNA, was characterized by its high phosphorus content and resistance to proteolysis, distinguishing it from proteins. DNA's role as the carrier of genetic information was established through pivotal experiments in the mid-20th century. In 1944, Oswald Avery, Colin MacLeod, and Maclyn McCarty demonstrated that DNA could transfer genetic traits between bacteria, suggesting its role as the genetic material. The elucidation of DNA structure in 1953 was a milestone in molecular biology. James Watson and Francis Crick proposed the double helix model, suggesting that DNA consists of two strands wound around each other, with complementary base pairing facilitating replication. Their model was informed by critical experimental data, notably Rosalind Franklin's X-ray crystallography and contributions by Maurice Wilkins, who collaborated with Franklin and shared her data with Watson and Crick, aiding their model development. In recognition of their contributions, Watson, Crick, and Wilkins were awarded the Nobel Prize in Physiology or Medicine in 1962. Sadly, Rosalind Franklin passed away in 1958 and was not eligible for the Nobel Prize, as it is not awarded posthumously. Within the cells of organisms, DNA is packaged into highly organized structures called chromosomes. Each chromosome consists of a single, continuous DNA molecule. Prokaryotes are simple organisms, such as bacteria, which typically have a single circular chromosome located in the cytoplasm, as they lack a defined nucleus. Eukaryotes, such as plants, animals, fungi, and protists, have multiple chromosomes contained within the nucleus of each cell. For example, humans have 23 pairs of chromosomes, while dogs have 39 pairs. This intricate arrangement is essential for the effective storage, regulation, and transmission of genetic information within cells, and is fundamental to the proper functioning and reproduction of living organisms. Gregor Mendel, an Austrian monk who experimented with thousands of pea plants from 1856 to 1863, is renowned as the father of genetics for his pioneering work on heredity. Through meticulous cross-pollination experiments with pea plants of the species Pisum sativum exhibiting distinct traits, such as flower color, pod texture, and seed shape, Mendel uncovered the fundamental principles governing the inheritance of traits. To understand these principles, we first need to understand alleles. An allele is a variant form of a gene located at a specific position, or locus, on a chromosome. Genes can have multiple alleles, each contributing to the diversity of traits observed within a population. In diploid organisms, or species with paired chromosomes, such as humans, individuals inherit two alleles for each gene, one from each parent. These alleles can be either identical, a situation referred to as homozygous, or different, a state called as heterozygous. The combination of alleles determines specific traits. For instance, in pea plants, the gene responsible for flower color has two alleles, one for purple flowers and one for white flowers. The interaction between these alleles dictates the plant's flower color. Some alleles are dominant, meaning their traits are expressed even if only one copy is present. Others are recessive and require two copies, that is, one from each parent, for the trait to be expressed. With that background, let's examine Mendel's principles. The law of segregation is one such principle which states that during the formation of gametes or sperm and egg cells, the two alleles for each gene separate, so each gamete receives only one allele. Upon fertilization, offspring inherit one allele from each parent, restoring the pair. Another of Mendel's principles, termed the law of independent assortment, proposes that alleles of different genes assort independently during gamete formation, leading to genetic variation. This means the inheritance of one trait, like seed shape, generally does not affect the inheritance of another trait like flower color. Although Mendel was unaware of the molecular basis of heredity, his findings laid the groundwork for our understanding of genetic inheritance. Mendel's hereditary factors correspond to what we now call genes, which are specific sequences of DNA that code for traits. Alleles are different versions of a gene that lead to variations in a trait. As we've seen, genes are located within DNA on chromosomes, which are structures within cells that organize and carry genetic information. Cell division in somatic or body cells occurs through a process called mitosis that preserves the standard number of chromosomes for an organism. Meiosis, on the other hand, is a specialized cell division process that reduces chromosome numbers by half to produce gametes, such as sperm and ovum cells. Was first described in sea urchin eggs by the German biologist Oskar Hertwig in 1876. This halving of the chromosome number is necessary so that the standard number of chromosomes is restored when a sperm and egg cell combine during fertilization. In 1883, a Belgian zoologist, Eduard van Beneden, expanded upon Hertwig's work by detailing chromosome movements during meiosis in Ascaris roundworm eggs, providing further insights into the process. A German biologist, August Weismann, recognized the significance of meiosis in maintaining chromosome numbers across generations. In 1890, he proposed that 2 cell divisions are necessary to transform 1 diploid cell into 4 haploid cells, ensuring genetic continuity. During meiosis, homologous chromosomes, which are pairs of chromosomes containing the same genes, but possibly different alleles, separate, ensuring that each gamete receives 1 chromosome from each pair. This process aligns with Mendel's law of segregation. In the context of evolution, DNA serves as the blueprint for constructing an organism and is copied from generation to generation, so that in general, offspring resemble their parents. Each strand of its double helix stores these instructions as a sequence of smaller constituent molecules, called nucleotides, of which there are 4 kinds, usually designated A, T, G, and C. This quaternary or 4-symbol code is conceptually similar to how computers store instructions using a 2-symbol binary code consisting of sequences of ones and zeros. And just as the symbolic binary ones and zeros can be physically manifested as tiny components of volatile computer memory chips that are either in a charged or uncharged state, the physical embodiment of the A, T, G, and C symbols are the nucleotide molecules, adenine, thymine, guanine, and cytosine. However, genes make up a relatively small part of the human genome. The largest part of our genome comprises regions that do not code for proteins. It was initially thought that these non-coding regions were non-functional junk DNA, but it has been shown that a small part of this non-coding DNA is responsible for regulating the expression of genes. So how does DNA build organisms? A simple explanation that omits many nuances is that there are sections of DNA called genes, which are transcribed into RNA, which is then translated into proteins. Some of these are structural proteins that serve as building blocks for cells, while others are functional proteins such as enzymes that catalyze biochemical reactions within cells, and some hormones. Thus, DNA ultimately serves as a template for building proteins, which in turn participate in building the organism. But sometimes changes in the DNA occur during the copying process. How is this important for evolution? There are 2 main parts to this story. Random mutations occur when changes in the DNA sequence or genotype of an organism introduce new genetic variations. In sexually reproducing species, such as Homo sapiens, the DNA changes that contribute to the evolutionary process are those that occur in the reproductive organs, specifically in the production of sperm and egg cells, as this is the only DNA that is transmitted from parents to offspring. While many such mutations are neutral or lethal, some can be beneficial and contribute to an evolutionary advantage. How this occurs is explained by the second part of the evolutionary mechanism. Non-random natural selection is a process whereby living things with traits or phenotypes that help them survive in their environment tend to live longer and are therefore able to reproduce. These helpful traits are then passed down to their offspring, becoming more common in the population over time. This means that organisms better suited to their environment are more likely to survive and reproduce. While mutation is a random process, natural selection is distinctly non-random and results from the interaction between the organism and its environment, which, in addition to natural forces, may consist of other organisms such as predators, prey, and potential mates. Metaphorically, you can think of this as the natural environment selecting which organisms will survive to reproductive age and pass their DNA on to future generations. A simple example is when animals with thick fur survive in a freezing environment to become potential ancestors for future generations, while those without such protection die out, removing them from the gene pool. Other examples of such adaptive advantages are better camouflage, faster running speed, and an enhanced ability to acquire food. Genetic drift is a mechanism of evolution that refers to random changes in the frequency of alleles, which are different versions of a gene within a population over time, which is not due to an adaptive advantage conferred by the gene or any of its alleles. Unlike natural selection, which involves differential survival and reproduction based on advantageous traits, genetic drift occurs when certain alleles become more or less common due to purely chance events, leading to significant genetic changes over generations, and is especially significant in small populations. One example of genetic drift is the bottleneck effect, in which a natural disaster, such as a flood or earthquake, drastically reduces the size of a population. For example, imagine a forest with a population of butterflies exhibiting a variety of wing colors that occur in more or less equal numbers. A sudden wildfire kills most of the butterflies, leaving only a small, random assortment of survivors. If the surviving group has a higher proportion of butterflies with blue wings, the allele for blue wing color will become more common in subsequent generations, not because it offers a survival advantage, but due to random chance event that increased its frequency. Then there is founder effect, in which a small group of individuals becomes isolated from a larger population and establishes a new colony. For example, consider a few individuals from a mainland bird population that are blown off course and settle on an isolated island. If these founding birds happen to carry alleles for unusually short beaks, the new island population may, over time, predominantly exhibit shorter beaks, regardless of whether this trait is advantageous. Gene flow, also known as migration, is the movement of individuals or the transfer of genetic material between separate populations. Such migration of individuals or genetic material can introduce new alleles into a population or alter existing allele frequencies. This movement introduces new genetic variations into a population, enhancing genetic diversity and potentially influencing evolutionary paths. This exchange of genetic material also tends to reduce genetic differences between populations. As an example, consider two distinct populations of wildflowers on opposite sides of a river, each with unique flower colors due to genetic differences. If pollen from one side is carried by wind or insects across the river to fertilize plants on the other side, this would be a gene flow event, and the resulting seeds will inherit genetic material from both populations. Over time, this cross-pollination can lead to a blending of flower colors in both populations, increasing genetic diversity and potentially introducing new traits. Non-random mating occurs when individuals in a population select mates based on specific traits or preferences, rather than mating randomly. A well-known example is that female peahens often select mates based on the size and brightness of the male peacock's tail feathers, favoring those with more elaborate displays. This selection can significantly influence the genetic structure of a population, such as increasing homozygosity or heterozygosity in the population, thus influencing allele frequencies. Some categories of non-random selective mating are inbreeding, outbreeding, and assortative mating. Inbreeding or mating between closely related individuals increases the chance of offspring inheriting identical alleles from both parents. An example of this is self-fertilization in certain plant species. Outbreeding or mating between unrelated or distantly related individuals, like when animals leave their birth group to join and mate with members of another group, promotes genetic diversity. Assortative mating has two flavors: positive assortative mating, in which individuals prefer mates with similar phenotypic traits to themselves, and negative assortative mating, also called disassortative mating, in which individuals prefer mates with different phenotypic traits to themselves. An example of positive assortative mating is tall people tending to marry other tall individuals. Examples of disassortative mating are where female mice prefer mates with different major histocompatibility complex genes detected through scent to enhance the immune diversity of their offspring, or where white-throated sparrows exhibit disassortative mating based on head stripe coloration, with individuals preferring mates with a different stripe color, thus maintaining genetic diversity within the population. The field of epigenetics brings us full circle to a reconsideration of ideas from the older, pre-Darwinian evolutionary theory, Lamarckism. The term epigenetics was coined by Conrad Hal Waddington, a British developmental biologist, in 1942. and is a field that examines how behaviors and environmental factors can cause changes in gene expression without altering the underlying DNA sequence. These changes can influence various biological processes and have significant implications for health and disease. While the conceptual foundations of epigenetics were established in the late 19th and mid-20th centuries, it emerged as a distinct and widely recognized field of study in the early 2000s, propelled by technological advancements that enabled detailed exploration of gene regulation mechanisms. Charles Darwin proposed that evolution occurs through natural selection, where heritable traits that enhance survival and reproduction become more common over generations. Epigenetics complements Darwin's theory by providing a molecular mechanism for how environmental factors can influence the expression of existing genes, potentially affecting an organism's phenotype and its evolutionary fitness. A well-understood type of epigenetic modification is DNA methylation, or the addition of a methyl group to specific parts of the DNA molecule. This generally causes gene silencing by preventing the binding of transcription factors and other proteins necessary for gene expression. Methylation can be transient and can change rapidly during the lifespan of a cell or organism, or it can be essentially permanent once set early in the development of the embryo. Other types of epigenetic modifications, such as histone protein alterations and non-coding RNAs, have also been studied in the context of gene regulation. Certain epigenetic modifications can be transmitted to offspring, a phenomenon known as transgenerational epigenetic inheritance. This process involves the passing of epigenetic marks, such as DNA methylation patterns or histone modifications, from parents to their progeny, potentially influencing gene expression and phenotypic traits in subsequent generations. The inheritance of epigenetic modifications suggests that environmental factors experienced by parents, such as diet, stress, or exposure to toxins, could potentially affect the health and development of their descendants. While these findings have given rise to a nuanced reconsideration of Lamarckian concepts, it is important to note that epigenetic changes are generally less stable across generations compared to genetic mutations. The stability and extent of transgenerational epigenetic inheritance in humans remain areas of active research. In mammals, while some studies have indicated the possibility of epigenetic inheritance, the mechanisms are not yet fully understood, and the phenomenon is not as well characterized as in plants and invertebrates. It's important to note that while epigenetic inheritance adds a layer of complexity to our understanding of heredity, it does not replace traditional genetic inheritance, but rather interacts with it, potentially influencing evolutionary processes and the expression of traits across generations. C.H. Waddington's 1957 work, The Strategy of the Genes, delves into the complexities of biological organization, development, and evolution, introducing concepts that have significantly influenced modern biology. Waddington visualized development as a ball rolling down a contoured epigenetic landscape where the valleys represent different developmental pathways. This metaphor illustrates how genes interact with environmental factors to determine cell fate during development. The landscape's topography reflects the regulatory networks guiding these processes. Imagine a stem cell at the top of the landscape. As it differentiates, it rolls down into one of the valleys, each leading to a specific cell fate, for example, muscle cell, nerve cell. The shape of the landscape, influenced by genetic and environmental factors, directs the cell's developmental trajectory. The concept of canalization describes an organism's ability to produce a consistent phenotype despite genetic or environmental variations. Waddington proposed that developmental pathways are canalized, meaning they are buffered against disturbances, ensuring stability in the organism's development. In fruit flies, despite genetic mutations or environmental changes, certain traits like wing vein patterns remain remarkably consistent, demonstrating canalization. Waddington suggested that traits initially produced in response to environmental stimuli could become genetically encoded over generations, even without the original environmental trigger. This idea bridges the gap between genetic determinism and environmental influences, highlighting the dynamic interplay between genes and the environment. Waddington exposed fruit fly embryos to heat shocks, inducing the development of an abnormal trait for example, cross-veinless wings. After several generations of selecting for this trait, it became expressed without the heat shock, indicating that the trait had become genetically assimilated. Waddington's work emphasized that development and evolution are not solely dictated by genes, but result from complex interactions between genetic information and environmental contexts. His ideas laid the foundation for the field of epigenetics, which studies how gene expression is regulated by factors other than changes in the DNA sequence. While Mendel's experiments with pea plants led to the formulation of the one gene, one trait model, suggesting that each gene directly corresponds to a specific trait. While this model was instrumental in the early understanding of genetics, it significantly oversimplifies the complexities of biological development and inheritance. C.H. Waddington's epigenetic landscape model provides a more nuanced perspective. He introduced the concept of the epigenetic landscape to illustrate how gene regulation during development is influenced by a network of genetic and environmental interactions. In this metaphor, a cell's developmental trajectory is represented by a ball rolling down a landscape of hills and valleys, where the topography is shaped by gene regulatory networks and external factors. A purely Mendelian view of gene action assumes genes act independently to determine traits. Waddington's perspective recognizes that genes interact in complex networks where multiple genes can influence a single trait and a single gene can affect multiple traits. The Mendelian view, coupled with a naive interpretation of Darwinism, largely ignores environmental factors in trait determination and can lead to the incorrect assumption that the only role of the environment is to select from the phenotypes which are entirely determined by the organism's genes. While Waddington's perspective emphasizes that environmental factors can alter the epigenetic landscape, affecting gene expression and consequently development, the Mendelian view suggests a direct, linear relationship between genes and traits. Waddington's perspective illustrates that development is a dynamic process with multiple potential outcomes, influenced by both genetic predispositions and environmental contexts. Consider the development of the nervous system. It's not governed by a single gene, but by a complex interplay of numerous genes and environmental signals. Waddington's landscape metaphor shows how various factors can channel the developmental process into different outcomes, even with the same genetic starting point. In summary, while Mendel's one gene, one trait model laid the foundation for genetics, Waddington's epigenetic landscape model reveals the intricate, multifactorial nature of development, where genes, their interactions, and environmental influences collectively shape an organism's traits. That ends this ThinkerFlix presentation on DNA, genetics, epigenetics, and the theory of evolution. Please subscribe to our channel and leave a comment.